Hello again, BIO178. In this last video here, we will talk about one of the reasons why it's actually, some people hesitate to use this technique of genetic rescue and translocations, and that's this idea of outbreeding depression. In other words, it's really important to think about which individuals you're moving between populations, and we'll talk about some reasons why in this video. So sometimes your intent can be to help and to move individuals in from a different population, but something called outbreeding depression can actually happen if the individuals you're moving are too dissimilar. They can actually produce offspring that have lower fitness in either parent. So again, this is kind of a Goldilocks type scenario where we've already talked about how you can have inbreeding depression if the parents are too related to each other. So in this hypothetical schematic, we have genetic distance between parents on the x-axis, the fitness of the progeny of the offspring on the y-axis. So if the parents are too similar, we see low fitness with inbreeding depression, but we also see really low fitness with outbreeding depression if the parents are too dissimilar. So we also want to make sure we're not going too far and moving individuals that are too dissimilar. In terms of why genetically this outbreeding depression might happen, there are a couple different plausible explanations. The first is that if you're moving individuals from somewhere else, this can actually lead to the loss of locally adapted genotypes. So we talked about this a little bit in the local adaptation lecture, and this is bringing this back in terms of why local adaptation, which is again the individuals that do better in their unique habitats compared to foreign non-native habitats. And that's important to think about in terms of conservation, because if you're moving individuals into a new area, they might not be adapted to that specific area. So the example I'm showing here is two different types of sticklebacks, and they live in different regions of the lake. So some live benthically on the bottom, some live limnetically, more on the top, and associated with these different habitats, they actually eat different things. And the different diet also leads to differences in their jaw structure. And what happens if you then cross individuals from these different types? So in their native habitat, they're locally adapted. They have the genes that give them the mouth morphology, for instance, to eat properly in these different habitats. So if we look in this graph on the left, we have the growth rate in the littoral zone, growth rate in open water, and we can see that the benthic species grows really well in its native habitat. Same for the limnetic species, grows really well in its native habitat. But when you cross them, their F1 hybrid doesn't really do well in either habitat. So this could be an example of outbreeding depression, where you're seeing now low growth in the offspring, lower fitness compared to either parent, because now it has this mix of genes and they're not really adapted to either one of these habitats. So you're losing locally adapted genotypes. Another really cool, well, not for this fish, but cool genetically reason for outbreeding depression is called the loss of positive epistatic interactions. So epistatic interactions are interactions between two different genes. And if that interaction between two different genes is needed or beneficial in some way, if you are moving individuals now from a different location and they're breeding with new individuals, you can end up losing that positive needed interaction. So in this example here on the right, we have two different types of swordfish, and there are two different genes we're considering here. One of these is considered a tumor gene, and one of them is considered a repressor gene. So if you have dominant copies of both the tumor and the repressor gene, you're fine. You're not going to develop any tumors. Similarly, if you don't have either of those genes, you're fine. So what happens here is when you have this breeding now of individuals that are genetically different, the F1 generation is okay. Both of those individuals are fine. But when you get to the F2, you now see these unique interactions that never happened before. So now you're losing this connection between the tumor gene and the repressor gene in some of these offspring. So specifically, in this individual here, it now has the tumor gene, but it doesn't also have the repressor. So you needed that interaction between both of those genes for this fish to be able to have high fitness and survive. And so what happens here when you have this outbreeding depression is you have this tumor gene but no repressor gene. So this fish actually has this overgrowth of tumors, which are shown here in black, because it lost that positive epistatic interaction with the repressor gene. And that was a result of breeding with another individual that was too genetically dissimilar. It led to outbreeding depression. So if we're thinking about, we probably want to have some kind of intervention to help the species, one of the things that conservation managers often consider is something called reintroduction. And reintroduction refers to releasing species on purpose, so it's intentional, into an area that was part of the historical range, but now they don't exist there anymore. So this is a graphic from the LA Times, I believe, for distribution of wolf. In red here is the current range, which is relatively limited, but in the tan color is their historic range, which was historically much, much wider. So reintroduction would be intentionally moving individuals into some of this historic range to move them to where they used to be. When we're thinking about which individuals to move and reintroduce into the area, genetic information can be really, really helpful and informative here in terms of choosing the correct individuals to make sure we're setting up these individuals for success. So an example of this was with oriental white storks, and they wanted to reestablish a population in Japan. And so one of the ways that you can try and figure out which individuals to move into the historic range that are most genetically similar to the individuals that used to live there is use museum specimens. So that's why there's this picture of this stuffed bird here on the right. So even if we don't have individuals living in that historic range anymore, there may be museum specimens that people have preserved from that historic range. You can take a piece of the foot or something like that, and you can actually get genetic data from those museum specimens, use that to identify the genotype of individuals that used to live in that historic range, and then try and match with your existing individuals from different locations and pick which individuals will be most appropriate to move into these what used to be occupied ranges for the species. So in terms of what techniques you use to actually match and identify appropriate individuals, you can use things we've talked about previously like haplotype networks and phylogenetic trees. So let's continue with this example with the Japanese stork. So say we have 3 different locations, right? We have Japan, China, and Russia. So China and Russia are live populations, and then the individuals from Japan, the only samples we have are museum specimens. What we're trying to do is figure out which one of these 2 populations to move into Japan to have a successful reintroduction. So what you do is take samples from each of those different locations, and then you could use a marker from the mitochondria, for instance, and you could use either haplotype networks or phylogenetic trees to figure out which of these live populations best matches the Japan population. So if we were doing a haplotype network, for instance, say this is one haplotype, and we found that there was a haplotype that was shared between Japan, China, and Russia. So say we have one haplotype that's found in all 3 of those locations, that would indicate that overall there's some genetic similarity there between both the China and Russia population, which might make it appropriate to move back into Japan for reintroduction. Similarly, if we were to use a phylogenetic tree approach, if we found that there was a branch on that tree that had individuals from Japan and China and Russia, remember, on a phylogenetic tree, individuals that are neighbors and are clustered together are genetically most similar. If we found that one haplotype was found across all of these sites and that a maximum likelihood phylogenetic tree showed no distinction across the three regions, we could then conclude that it would be appropriate to move birds from either China or Russia back into the historic range of Japan, because both individuals from both of those regions seem to match the genotypes that historically were in those ranges. So that's how we can use haplotype networks and phylogenetic trees to help us inform conservation management. Another thing we can use genetic information for is to help us make sure that this new population we're establishing has enough genetic diversity. So remember, this is important because in order to have a reasonable probability of adaptive potential and to avoid potentially deleterious effects of inbreeding and genetic drift, we want to make sure we have appropriate genetic diversity. So essentially this founder population that we're moving, that we're translocating, we want to make sure that they have high genetic diversity. So an example of this is with this bird, a lot of birds in this lecture, called the northern bald ibis. And so this is a bird that currently is only found in the wild in Morocco. My geography is terrible. I had to look up several different maps to make sure I knew what these different countries were. So this is the current live population. They also have a population in Turkey that was reintroduced. So it went extinct there. They used some individuals to reintroduce them into Turkey. And so when they're trying to figure out now what individuals they should use for reintroductions and whether they have appropriate genetic diversity, one of the things they're considering now is whether individuals they have in a zoo population contain sufficient genetic diversity to be able to have them be reintroduced and to have that population be successful. So in this table, what's being shown is they're comparing two different populations, a zoo population and this other population here that was used for a successful reintroduction into Turkey. And so what they found is that overall, the genetic diversity of these two populations, the zoo population and the population that was used for reintroduction to Turkey, have roughly comparable genetic diversity. So they concluded in this paper that this zoo population is indeed appropriate to use for reintroductions because it has as much genetic diversity as other individuals that have been used to have successful reintroductions in other regions. So if we're thinking about other potential threats or conservation things to consider, one of the things that's often talked about is that hybridization with other species is a threat to a pure species. So here in this graphic here, we have two different species, a green one and a red one. And so what this means, being a threat to a pure species, it can lead to something called genetic swamping, where essentially if we initially have a hybrid being formed that's half green, half red, if that hybrid keeps mating with an individual from the green population, eventually what's going to happen is you're going to end up with an individual that has a mostly green genotype, and the red genotype has essentially been lost. So you've lost that quote-unquote pure species because of this genetic swamping from the green species. So it's something that conservation managers try to consider, is whether hybridization can basically lead to the loss of a pure genetic species. On the other hand, though, hybridization can be beneficial if it leads to the introgression of adaptive genes. So in this schematic shown here, we have introgression. So the black regions are introgressed from this donor population into this recipient population. Overall, introgression is relatively rare, but if it's adaptive introgression, that actually means it's going to be more widespread in the population because it's beneficial. So selection is going to act to have it become more prominent in the population. So in this case, hybridization, introgression of genes from one population to another can actually be helpful and help that population survive. An example of this is with two different species of butterfly. The butterfly here on the left has what's called that warning coloration. So the coloration pattern tells predators, I don't taste good, I'm poisonous, don't eat me. And so what's happened is genes from that species have actually repeatedly introgressed into this other butterfly species because this coloration pattern is beneficial. So if you're telling predators not to eat you, that's going to help you survive. So this is an example of introgression of adaptive alleles that encode that warning coloration. And lastly, just to mention that there's another growing subfield in conservation genetics that's called community genetics. And again, just so you know this exists, if you hear about it or see it somewhere, here the focus is linking genetic diversity not just of the species itself, but also to how the broader ecosystem around it functions. So in this image that's shown here, overall what we're looking at here, the interpretation is similar to a PCA. So here the different colors represent different genotypes of plants. And what we can see is that these different genotypes of plants actually lead to a different variety of arthropods on the plants. So that's what's indicated here by the clustering of these different samples. So we can see that for these two genotypes, for example, they don't overlap. So what that means is that when you take into account the different arthropod species found on these different genotypes of plant, even though it's the same species of plant, the different genotype actually leads to a different community assemblage of arthropods on that plant. So it's a growing field now with looking at this connection between genetic diversity and ecosystem functioning.